Today's webinar presentation is titled White Pine, Updates on Pests and Diseases Affecting White Pine in the Northeast. Our presenter today is Isabel Munck. Isabel has served as a forest pathologist for the USDA Forest Service since 2010. She provides assistance to state and federal partners on all aspects of forest pathology and management. Isabel has a PhD in plant pathology from the University of Wisconsin-Madison, a master's in forest pathology and mycology from SUNY ESF, and a BS in forest management from Colorado State University. And with that, Isabel, welcome, and thank you for the presentation today, and I'll go ahead and turn the reins over to you.
Sure. Thank you, Kate, and welcome everyone. Today I will be presenting work which I have conducted, but also work from two graduate students, Cameron McIntyre from UNH and Kara Constanza from UMaine. Both Cameron and Kara were conducting their PhDs, and now Kara is a professor at University of New Brunswick. Okay, so moving on to the next slide. Hold on, there we go. So all our work is extremely collaborative. We work closely with departments of forest health, each for every state in New England, and with many folks from many universities. So, and also from like the Natural Resource Conservation Service. So I just want to acknowledge all the many people that have helped us. And as you know, white pine is an important resource in New England. It's in New York. It's not only important ecologically, but it's very important for the lumber and forestry industry. White pine represents the greatest portion of growing stock in Massachusetts and New Hampshire, 24% for Massachusetts and 21% for New Hampshire, and then 9% for Maine. And Maine is the #1 white pine lumber producing state in the nation. And today I'll be talking about 3 different topics: foliar diseases of white pine, Caliciopsis, canker, and white pine blister rust. After each section, I will stop for a minute so that I may answer any questions that might arise from that section. And then there'll be also a answer question section at the end. So the first topic are foliar diseases of white pine. We call these white pine needle damage. This was one of the first things I started working on when I started my job at the Forest Service here. Aerial surveyors were detecting needle, like foliar discoloration, so yellowing of the crowns or browning of the crowns. And every year, each state and also the Forest Service conducts aerial surveys to see if there's any damage causing mortality or defoliation or discoloration of the foliage. And during these surveys, white pine seemed to be affected by something, and it was unclear what was causing the damage. So we were tasked to try to figure out what was causing this discoloration of white pine. And this is a map from aerial surveys conducted in 2017, so last year. And typically these aerial surveys are conducted later in the season, in the field season, so more like around August. But last year, all our state partners flew a little bit earlier in June and July in order to capture this discoloration of white pine, and they mapped 95,000 acres. The symptoms that we see are either browning of the needles, as seen in these trees, or yellowing, and all age classes are affected, mature trees and region as well. Here's a better picture of, let's see if I can use my pointer. So this is a row of trees in Maine, and you can see this white pine. Sorry about that. This white pine in the middle is yellow, whereas other trees are greener. Apologize, I'm trying to transitions, and oh, here we go. Okay, and so the foliage that's affected are last year's foliage. This is a picture of a tree. You can see the new candles it's putting out. Those are not affected. It's last year's foliage that's affected. And actually the New Year's foliage does get infected this year, but the symptoms won't show until the following year. The needles turn yellow or brown starting in May to early June, July, and then they fall off. So once the needles fall, it's really hard to detect any damage from aerial surveys because most of the damage is on the bottom 2/3 of the crown. And when they're flying over the pines, the very tops look green. It's very hard to detect this damage. So here's a close-up of a tree that's been severely defoliated, and you can see it's only holding current year needles where it should be holding at least 3 years worth of needles. And defoliation in conifers is very detrimental to their health because conifers hold most of their starches and storage materials in their needles, as opposed to hardwoods that hold a lot of those materials in their roots. So whereas hardwoods that are defoliated can flush out again after they're defoliated, conifers cannot. So the first thing we wanted to do was just monitor the long-term effects of this defoliations on the pine. And to do this, we set up plots, permanent monitoring plots in 8 locations throughout New England. And every year we go out and look at the trees and rate them for damage. And then we wanted to see the impact of the foliation on growth, so we also took cores from each tree. So this is a map of all our long-term monitoring plots. 2 of them are in Maine, 1 of them is in New Hampshire, 1 of them is in Massachusetts, but most of them are in Vermont. And at each of these locations, we tagged trees that had crowns that were affected by the needle damage, such as this one, and also nearby trees that were healthy, like this one. You can see that the damage can vary by stand. You can have trees that are healthy and some that are not. And we have been monitoring these trees for many years now, so 2012. And what I want to point out is these graphs is chlorosis, or yellowing on the left, defoliation on the right. The black bars represent trees that were initially unhealthy, and the gray bars represent trees that were initially healthy. You can see that trees that were initially healthy are starting to show symptoms, and both chlorosis and defoliation, and the severity of the symptoms is increasing. On the y-axis is the proportion of the crown that's affected. So bottom third, damage limited to just the bottom third, more than 2/3 of the crown more than 1/3 of the crown affected, and more than 2/3 of the crown affected. So the severity of the symptoms seems to be increasing in both the trees that were initially healthy and the trees that were already showing symptoms in 2012. And we collected thousands of needles from many symptomatic trees throughout New England and New York, and there's not one specific pathogen that's causing these symptoms. There's several. All of these are foliar pathogens, and the most prevalent one is this one, Lecanosticta acicola. This is the pathogen that causes brown spot needle blight. Now, brown spot needle blight was a problem for Christmas tree growers in the Midwest and the South, but this type of damage on mature pines is unprecedented. And all these needle pathogens have similar life cycle. Their rain splash is first, so usually the bottom part of the crown is affected the most because the spores spread from higher parts of the tree to the bottom part of the tree. And needles are affected in their first year when those candles are elongating, but then it takes an entire year for the symptoms to appear in the second year needles. And here's a diagram of one of these pathogens. Actually, this is the Lecanosticta acicola, so the brown spot needle pathogen. You can see the spore lands on a needle, and then it grows through the stomata, through the living tissues of the cell, for an entire year it does this, and then it ruptures on the other side the following year. So we did some modeling to see what was going on, why were these pathogens all of a sudden a problem. And what we found is that there's been a long-term increase in summer precipitation throughout time. There's a greater amount of precipitation that's falling in New England, and that's falling in the summer and the spring. And that greater amount of humidity and precipitation favors spore production and dissemination of all these pathogens. Specifically precipitation in May. And it's got a 1-year lag. So if it's really rainy and wet this spring, you won't see, and which favoring the reproduction and dispersal of these pathogens, you won't see the symptoms until the following year. And now so that took a lot of effort trying to figure out what was happening with the timing on the different fungi. But what I'd like to focus on today is what it's doing, what these pathogens are doing to the trees, and also what management alternatives we can take to mitigate the damage. And this work was done by Cameron McIntyre. You can see here on the right. So Cameron went out and collected foliage throughout the from starting in May to October at 4 locations. These are throughout New England, so some in Maine, some in Vermont. And what you're seeing here is the amount of foliar biomass that he collected in his traps on the y-axis, and then through time on the x-axis. Now, pines typically the foliate in October. So the bar represented here, the October bar, would be normal, the time when trees would normally shed their needles. So what I want you to see is that these pines, and then another thing I want to point out is May would be the time when pines are not shedding their needles. So that's kind of like the background amount of needles that are shed by pines. And in these trees that are affected by these foliar pathogens, a lot of them are shedding their needles in June and July, to the point at this site, for example, that's very severely impacted. They're shedding more needles in June and July than they are shedding in October when they're supposed to be shedding their needles. So these numbers on the top represent the proportion of their needles that the pines are shedding in June and July versus October. So this site in Durham is kind of the control site where there's the least amount of damage, but still 39% of the needles are being shed in June and July, versus the site in Bethel that has a lot of symptoms and 68% of their needles are being shed in June and July. And what this defoliation is doing is it's impacting the tree's ability to put out growth. So this is showing up in the dendrochronological analyses. The foliar damage started being apparent in 2009, and you can see that this pine was growing well up to this point, and there's big reduction in growth ever since it was hit by the foliar pathogens. And so Cameron has been quantifying the amount of decline in basal area that trees are putting out in several locations. And this is what this graph is showing. Each panel is for a different location, and the black lines represent the basal area increment here on the y-axis for the trees that we tagged that were initially healthy. And the red lines are the basal area increments for the trees that were initially unhealthy when we started doing the ratings for disease damage. So for all these sites, or for most of them, you can see that the growth of these two types of trees was pretty similar, although the trees that were initially damaged by white pine needle damage had slower growth rates than the trees that looked healthy. But starting in 2009, there's a divergence in their growth. So the trees that were initially healthy are growing well, and the ones that were damaged in 2012 are showing a decline. And some of these sites were old growth. So let's see, Mohawk, so this site, Mohawk, this is the Mohawk Trail in Massachusetts. These trees are hundreds of years old, and they were growing very well until they were hit by these needle pathogens. So this graph is a combination of the information from all those graphs. So the trees that were initially showing low severity in 2012 haven't really changed in their mean basal area, whereas the trees that were affected have shown a decline in growth. So we wanted to know if there's anything we could do to mitigate this damage that we're seeing, and what we did is we set up an experiment in 2 locations, both of them in New Hampshire, where Cameron went out and looked at the crown ratings for all the trees and rated them based on disease severity, and then they implemented thinnings. So they did a low density thinning, a high density thinning, and a control, and these were replicated at both locations. So this is 2 control plots for the Fox location, 2 high density thinnings and 2 low density thinnings. And he went out and collected data at post, before thinning, and then after thinning, and then 2 years after thinning. So what I'm showing you here is the white pine needle damage severity ratings. 0 would be no disease, and 2 would be more than 2/3 of the crown are affected by damage. And this is the year. So before any of the treatments occurred, there was no difference amongst the sites. And then again, the year after the thinning, there's very little difference because it takes at least 1 year for the symptoms to appear, plus it takes some time for the effects of thinning to manifest themselves. But then by 2017, the crown ratings for the site where no thinning occurred were greater. So there was more damage in the control site compared to the 2 other sites that were thinned. So thinning is ameliorating the effects of these needle pathogens, probably because by reducing competition in the stand, the trees that are residuals are better able to put up with the defoliation, and also because the pathogen doesn't spread very far. So by increasing the amount of space between trees, you're reducing the likelihood that the pathogen will infect the neighbors. So the major findings that we had were that this foliar fungi are causing, at least 40 to 78% of the total annual litter fall when untimely. This is untimely. This is at a time when the trees need to photosynthesize. And the foliage that remains after the foliation is only partially developed, so those candles take a long time to flush out and are not able to make up for the fact that the older needles have fallen. And the foliation is causing about 40% decline in basal area, but thinning and low density stands can increase tree vigor and mitigate the effects of white pine needle damage. So at this time, I like to pause and see if there's any questions on this section.
Thanks, Isabel. There is one question from Harris. Is there a finer scale map of the white pine needle disease distribution in Vermont? If, for example, it would be available to check against individual properties.
No, there is not. We are working with remote sensing specialists to see if we can use the foliar flight data to then verify, use monitoring like a satellite data and the flight data to then do an extraction to have better idea of where it's occurring. But we're still working on that. There's really no product that's available yet.
Okay, thanks. And a quick question from Andrew, talking about the reasons that you gave for low and high density thinnings that were successful. He's wondering, could also the fact that the thin stands have better airflow and dry out more quickly influence the success?
Right. Yes. So the So I think the reason why their success is because conditions improve for the tree. Also, the pathogen cannot disperse to its neighbors, and as Andrew pointed out, there's less humidity and rain. Or the humidity and rain as they hit one tree cannot reach the neighbors. So yes.
Okay.
We haven't
Great.
tested that, but that is our hypothesis.
Makes sense. That's all the questions that we have for now. So if you want to move on to the next topic,
Sure.
that'd be great. Thanks.
So moving on to Caliciopsis. So Caliciopsis canker was reported back in the 1930s. Oh, I forgot to mention the needle pathogens are native. It just seems that the weather conditions have improved their, have caused their current epidemic. Likewise, Caliciopsis canker is also native. It was first reported at the late 1800s and was very common in the 1930s. And then all of a sudden, there's no reports of it. We think it's because people were so focused on white pine blister rust that they kind of forgot about this. But recently, there's been an increase in reports throughout the range of white pine from the Midwest to the South. And also it is reported in native and exotic pines planted in Europe. The fungus is allegedly, we know very little about how this fungus works, but allegedly the fungus attacks thin-barked areas of the bole and the branches. And again, all size classes are affected. So Caliciopsis is a fungus. It's an ascomycete. It causes cankers. You can see it with your naked eye. The fruiting structures look like eyelashes. It is more severe in pole-sized stands than in regen than in mature trees. Mature trees usually show damage at the very top of the crown of the branches, not in the bole. This is a pole-sized stand, and you can see there's a lot of resin coming from these trees, and it's throughout the entire bole. It doesn't seem to be coming from a single source. It's spread throughout the entire bole. Those very heavy resinosis occurs when the cankers are active. Later, as time goes by, you can see that these cankers kind of close. The resin isn't as white, it's more dark, so they're harder to see. But if you were to peel back the bark from these cankers, there's damage to the sapwood of the tree. Sometimes the tree is not able to close these cankers and compartmentalize the damage, and then you can have more severe symptoms. So we were worried about the ability of this fungus to degrade the wood product. So this work was done by Cara Constanza at UMaine, and she had the two objectives from her work I'm going to be focusing on today are ecological and economic. And she wanted to know she wanted to section the trees where the cankers were occurring to try to see if we could figure out what was causing this damage, with the dendrochronological work, and also try to quantify, was this actually damaging the product? Was it causing damage that was quantifiable economically? And actually, when I was putting this talk together, I didn't have time to show my own work, but we did a big survey throughout New England and found Caliciopsis canker everywhere. It's very widespread, but it's more severe in soil types that are excessively drained, where trees are more likely to suffer from drought stress. So within the range of stands that were affected, we found 3 sites that had Caliciopsis canker. So all these sites were very damaged by Caliciopsis. And then there was a thinning that was conducted 5 to 15 years ago to try to ameliorate the damage from Caliciopsis. So we wanted to see if this thinning was successful. So we rated the trees by the external symptoms to see if they were severely damaged by Caliciopsis or not. And then these trees were cut after we rated them for external symptoms. They were cut in sections. So every foot, Cara cut out a cookie to count the number of cankers that were present, and how wide they were, and so forth. It's very labor intensive. And she did this for 36 trees, so it was like a tree dissection. Okay. She also, I don't want to talk about this, but she made sure that the pathogens that we thought were involved, and did a lot of work in the lab to make sure that that was happening. She also felled another additional 60 trees to see, it was like a mill study trying to see if it was affecting what the economic impact of this canker was at these sites. So these were graded. This was all done commercial, like by as if they were treated as commercial lumber. And she quantified the grade and financial loss in these trees. So what I want to show you is, like I mentioned earlier, what we think is going on is that trees with the thinner bark, so pole-sized trees or the upper part of the crown are most likely to be affected. And she wanted to see if the butt lock was unaffected. So in this graph, we're showing the number of cankers from 0 to 21. And the x-axis is, oh wait, I'm sorry. The number of cankers is on the x-axis and the canker height is on the y-axis. So what she's showing is that at this mid-height of 9 to 12 meters, so in the middle of the bole of the tree is where you see the most cankers. The butt log, which would be here, there's not as many cankers, which is good in terms of lumber value. And that's what she's showing you. The line where damage is that line is the butt log. So most of the damage is above the butt log in this section. And this is a very busy graph, but all I want to show you here is that all these variables are related to tree health. So crown ratio, you want the bigger crown ratio, the better it is for the tree. You want a more, hopefully 50% or 40% crown ratio. Crown density, sapwood area, basal area, all these things, the more a tree has, the better, the more healthy it is. And what these graphs are showing is that trees are affected by Caliciopsis. There's a downward trend in the crown ratio, in the crown density, in the sapwood area, and the basal area. So all these measurements of tree health are declining in relation to presence of Caliciopsis canker. And then she looked at those cookies she took from those trees that she felled and cut into sections, and she did dendrochronology to see, when these cankers started to occur. So percent of cankers per year from the cookies. And what she found is that there was some incidence of Caliciopsis dating back to even 1968. But after 1995, the number of cankers greatly increased. And it all started after a drought in 1995. So a lot of canker pathogens act this way. They affect trees that are stressed. So the idea is that these stress trees from the drought, were susceptible to the canker, and then there's a much greater incidence of canker after 1995. And then so that was all the ecological work. For the economic studies, is what they did is they processed these trees, and then a grader came and looked at the trees and determined, what the damage was from the defect. And then they kind of closed their eyes and graded the log as if it didn't have the board, sorry, as if it didn't have a defect, to try to determine, how much damage this canker was causing. So this is the type of damage that Caliciopsis causes to lumber. So as I said, they would grade it with the damage, and then they would block that out and grade it again. And this is on the y-axis, the percent of boards that were graded, and then on the x-axis in thin, and then thin stands for each of the sites. So it doesn't. So basically, what she wants to show is that damage was usually limited to 10% of the boards, or 10. Here, this site has the most, almost 20%. But some of the boards in this brown, less dark brown color, did have damage from Caliciopsis, but they were not downgraded. And when the boards that were downgraded only lost 1 grade, so this is all good news. It wasn't causing only 20% of the boards and sites that are really affected by Caliciopsis, and not a huge loss in grade. Another thing she did is quantify the amount of defects, other defects that were causing damage. So in this color, this is the amount of damage that was caused by red knots, and this is the greatest, whether the sites were unthinned or not, showing here in the y-axis, in the x-axis, non-thin, or this is one site thinned, one site unthinned, same site unthinned. The defect that was causing the most damage were red knots, and not Caliciopsis. But for the boards that were damaged by Caliciopsis, shown in black, those showed greater amount of biotic damage compared to boards that were not damaged by Caliciopsis. And that's what that's showing you, that arrow. There's greater amount of biotic damage on boards damaged by Caliciopsis. So it didn't look like there was a big difference between thin and unthinned sites, but these sites were already damaged by Caliciopsis, but when they were thinned. And I don't have time to show you my own work, but what I found is Caliciopsis incidence is greater in sites that were overstocked compared to understock. So I do think that maintaining proper stocking would help mitigate, at least prevent trees from having Caliciopsis damage. And the damage was about 2%. The percent value lost by C. pinea was about 2%. And when she talked to people that own the mills and are concerned about economic impact, they're worried at 1% is something that they're concerned about. So greater than it is causing damage, but not as much as you would think. And most of the damage is concentrated in the top part of the tree. So in conclusion, canker damage is synchronized among sites, meaning like she saw the same trends with the timing of the cankers, as starting after 1995 with greater incidence. And cankers are associated with factors measurements of tree health, such as crown ratios. Cankers seem to be associated with drought and incidence of hurricanes. So canker pathogens usually enter the tree through wounds. So hurricanes would cause wounds in trees that would be exploited by canker pathogens such as Caliciopsis. For lumber economic impact studies, she found that 39% of the lumber in sites where C. pinea was a problem, 39% of the lumber was affected, 11% of the lumber was downgraded, but Caliciopsis canker was not the greatest source of defects. Red knots were, and on average it causes 2% value loss. So, with that concludes my Caliciopsis section. And at this point, I'd like to pause to see if there's any questions about this.
It looks like we have a question from Earl. What is the measure of thinning? Is it residual basal area or crown separation?
I think it was residual basal area. So these were stands. Most of them were in New Hampshire, and they were managed by the state. And I think the residual basal area was 60 square feet per acre, but I'm not 100% sure.
Okay, great. Well, that's it for questions at this time.
Okay. So moving on, this is my last section. It's about white pine blister rust. White pine blister rust is caused by an exotic pathogen. So the other two issues that I talked about were native pathogens. This is an exotic. It's a fungus. It was introduced to North America from Europe at the time. I mean, back in the day, in the early 1900s, we had such a need for seedlings because there were so many huge reforestation projects that our native nurseries did not have the capacity to fill that need, and so seedlings were important from Europe. Now, this white pine is an American species, so it was grown commercially in Europe. So the seeds were shipped to Europe, and then it was grown there, and then was shipped back. And in these live tree shipments is how the fungus was introduced, although it's native from Asia, so it's also exotic in Europe. It has a very complicated life cycle. It has 5 spore stages, and it needs at least 2 hosts to complete the life cycle. So 5 needle pines and plants in the genus Ribes, although now we know that there's other plants that also are alternate host. So the spores in the pine, spores are produced, what we call the blister, it's called blister rust because of these blisters that are formed, and these are formed in the spring. And these are ascospores. So these type of spores that are produced in the spring infect the ribes in the spring, and then the fungus grows through the ribes leaves, and then at the end of the summer, it produces masses of spores on the ribes leaves that then later on in the fall infect the pine. So as I mentioned earlier, the fungus was introduced in the 1890s, and then it spread quickly throughout the Northeast. At the time, there was a lot of regenerating white pine, and that was the size class that's most susceptible. Because of this disease, it was the beginning of federal quarantines and plant regulation in this country. This happened right after the chestnut blight, and people were really worried about it, and so they banned importation and importation of white pine from Europe, and also cultivation of ribes. A lot of money was spent on eradicating ribes. If you remove the alternate host, this breaks the disease cycle. So the fungus cannot complete its life cycle without the alternate host. So if you remove the alternate host, it breaks the disease cycle. And a lot of money was spent by the Civilian Conservation Corps and different state agencies and the federal government to eradicate the disease. Now, eradication worked in the East because we have a lot of roads. Our native ribes are not as susceptible. The terrain is more accessible, and so it did work in the East, but it did not work in the West. So currently, we have ribes restrictions in Maine, New Hampshire, and Massachusetts. There's no restrictions in Vermont, Connecticut, and Pennsylvania. And so I'm showing you statistics for New Hampshire because I'm going to be describing a study we did in New Hampshire. In New Hampshire, again, the disease was introduced same time after New York, and in the late 1900s, and mortality rates were huge in the beginning of the 19th century. In some locations, up to 50% of the pines had died. And ribes in New Hampshire were eradicated first through federal programs and then by the state until 1986. Then there was a big push from the juicing industry who wanted to grow ribes because ribes is high in antioxidants and people want to use it to make wine. So there was a big push to try to grow ribes commercially, and new varieties of ribes that were resistant and supposedly immune were available for planting in the 1990s. So in the 1990s, the state of New Hampshire decided to see what was the current status of white pine blister rust, and they conducted a huge survey statewide, and they found that blister rust incidence on the pine was pretty low, 3 to 7%. So they allowed ribes to be planted, and only varieties that were tested in field trials in Oregon, where they grow ribes commercially, to be resistant. So they only allowed currants that were or gooseberries that in field trials appear to be resistant. Okay. So in 2011, right after I started my job, I read this article where they showed that white pine blister rust was found on immune black currants. So the biggest there's a lot of ribes, and some of them are native and some of them are not. Now, black currants, which are European, are the most susceptible, and they produce more spores, better spores than any other currants. And so what this article is saying is that these black currants that had one gene that allowed them to be resistant to disease were no longer resistant. And this was found in Connecticut. So we want to know that if we were, what was happening in New Hampshire, were the ribes that were planted in New Hampshire also showing this break in resistance, and we just had to wanted to see also what happened after all these years where these ribes were planted. And I want to say to do our study, we used these permits that the state of New Hampshire, if you want to plant ribes in the state of New Hampshire, you have to apply for a permit. And so you have to state like what ribes you're planted, and you can only plant a certain amount of ribes that the state allows, as I mentioned earlier, that are not supposed to be susceptible to blister rust. So we visited every location for someone that had applied for a permit, as shown on this map. And all the sites with an X are sites that were positive for blister rust on the ribes. So most sites look like this. They were private gardens, a lot of very good gardeners in New Hampshire, but white pine is very common, so you have the ribes in the forefront, and then you have white pines in the background. We did also visit some commercial plantings, as shown here. Again, you have ribes in the forefront, and then white pines in the background. So at each location, we looked at least 5 plants of each cultivar. Cultivar just means or cultivate variety, so not necessarily species, but different types of ribes. So European black currants of different cultivars, and some of them were resistant. Like Titania, for example, is a cultivar of European black currant that contained this gene that was conferring it resistance, and now the fungus is able to overcome. So we looked at a lot of plants and several leaves per plant, and made sure that what we were looking at was blister rust by sending samples to Cornell for molecular analyses. And then we also sent samples. One of the things that we got pushback on is like, well, maybe the plants that you're looking at that you think are resistant are not actually resistant because they're not what you think they are, meaning there's some kind of contamination at the nursery. So they were propagating what they were saying was Titania, perhaps, and it wasn't really Titania. So we sent samples to Canada where they have the original cultivars from their cultivar collection, and we sent rust samples from New Hampshire that they could then infect their original cultivars to make sure that we weren't seeing a supposed break in resistance because of contamination of the ribes at the nursery. What I'm showing you here is a probability of blister rust, so it's almost like incidence from 0 to 100% of the different types of currants. So the first bars are currants that had this gene that supposed to make them completely resistant to the disease. We did not find that they were. We found that 30% of the time they had blister rust. And then all these other types of varieties of currants that are allowed for planting in New Hampshire. Interestingly, the red and the white currants were the ones that were least likely to have blister rust. So only about 20% of those plants had blister rust. We also looked at the disease severity, so what proportion of the ribes leaf was affected. And that's what I'm showing here on the y-axis, from 0 to 18%. So those of the plants that were infected, the European black currants with that resistance gene actually had the greatest proportion of the leaf that was affected. So in the literature, the European blackcurrants are described as spore factories, and that's what we found in the field. That once the resistance was broken, these plants were making a lot of spores. They have bigger leaves, and then a greater proportion of the area of the leaf was affected by blister rust. So at these sites, we wanted to see if the pines that were surrounding the locations where the ribes were planted were affected. So we looked at 12 pines closest to the ribes planting in each cardinal direction up to 1,000 feet. And we recorded their size and their distance from the ribes and whether they had blister rust or not. So these are the symptoms on the pines. You see the axial scars left over from the acia that are put out in the spring. Also resinosis, but unlike Caliciopsis that's spread out throughout the entirety of the bowl, usually blister rust is at one location. The source of the resinosis is from one canker, one location. So this graph is showing the incidence of blister rust in the pines. And what we found is that the pines that were planted close to the European blackcurrants with the CR gene were more likely to be affected by blister rust than pines that didn't have that pines planted next to currants that didn't have blister rust. So the black bars are data for pines that had acia. So for sure we know those had blister rust, and the hash bars had other symptoms of blister rust like the resinosis. So basically, the black bars are more conservative estimate of whether the pine had ripe blister rust or not. And the trend is the same. Basically, the pines planted next to the currants with a CR gene that now is no longer conferring resistant were the ones that were more likely to be infected. And we also measured the size. So seedlings are more likely to be affected than saw timber. The blister rust incidence on the y-axis decreased with tree size on the x-axis. So in conclusion, we found blister rust on all the cultivars that were, these cultivars in Oregon did not have blister rust, but they were in the field here in New Hampshire, they did. And Titania, which is a European blackcurrant with a CR gene that is no longer conferring resistance, were the ones that were that was the most frequently planted cultivar, because European blackcurrants, for people that like currants, are the nicest. And they were supposed to be resistant, and they're not. And pines that are neighboring these plantations of, or not plantations, but plantings of currants are more likely to be infected. So basically what the state did is they removed the European blackcurrants from the list. They can still plant other currants, but not the European blackcurrants. The recommendations for blister rust control haven't changed. These have been in place for a century. Basically what's recommended is pruning the lower and infected branches, removing infected pine trees, removing currants, and now there's resistant varieties of white pine that are available that are coming out of the Midwest. So with that, I'd like to thank you for your attention. I'd like to acknowledge the rest of the people that I didn't acknowledge in the beginning. So many people have helped us, including all the homeowners that allowed us to look at their currants in their properties. And here's my contact information.
Great. Thank you so much, Isabel, for your presentation today. So folks, if you have any questions, we have a few minutes, so please go ahead and type your questions into the question box on the side panel. And we'll give folks a few minutes to send in some questions. While we're waiting for questions to come in, just wanted to share with webinar participants today that our next webinar will be on March 1st, and we'll be hearing from Amanda Bruntz with the University of Connecticut on tree biomechanics. So if you haven't signed up for that webinar, please go to the webinar series webpage at rvermontwoods.org/forestry webinars and sign up for our next webinar in March. Okay, so we have a question here. I actually think that's one that you've already answered, Isabel.
Okay.
Hold on 2
And
seconds here. Looks like there is another question. Sorry. I guess a comment from Dave and maybe a question here. Thank you. Excellent presentation. Sorry to be a bit off topic, but we're seeing a great increase in foliar and twig diseases on red pine. Is there anybody in the region seeing this, and is there anybody investigating?
yes, it is region wide, and there is someone that's investigating it. His name is Michael Simmons. He's a PhD student at UNH. It depends on the site. It's not what the causal agents are not consistent. What's consistent is that red pine is kind of crapping out throughout the region, and then sometimes it's dying. Depending on location, it can be the red pine scale. Other times it's diplodia canker, which is different from Caliciopsis, but similar in its biology. And he will be finishing his PhD this soon, so hopefully we'll have more information we can share.
Great, thanks for sharing that, Isabel. And maybe I'll follow up with him and see if he or somebody else from his team would be interested in doing a webinar. Because I too have heard, of a number of the issues of red pine in the region. Well, great. Well, I don't see any other questions coming in, so it sounds like you covered the topic thoroughly. And I really, Isabel, I really appreciate your time this morning and sharing all of your research. And I know it's not necessarily a positive story, but it's helpful for you to share some of your management strategies and techniques to help address all three of these pests and diseases on the landscape. So.
Well, I hope I've shown you that management does work if it's implemented. Also, I wanted to mention we did our New Hampshire, the currants, the white pine blister rust study was done in New Hampshire, but Vermont allows planting of currants, and Dr. Dale Bergdahl from UVM had done studies before he retired showing the same trends in Vermont.
I was going to ask you, sort of, what the update is, and sort of, it sounds like in New Hampshire that you've been able to work to get the European rabies taken off the permit list. Has there been any conversations around kind of the implications of the amount of rabies out on the landscape now, or in terms of
Well, I
policy
think
making?
the reason we don't have blister rust issues is because we're writing on the success of our history. There was so much effort to remove rabies from the landscape that it worked, and we know this from, like in Maine, they showed that in locations where they had implemented the rabies control, there was less incidence in the white pine than in other areas where they had not done the rabies control, and so we know that it worked. And so because of the success, there was people forgot about blister rust, and several states were thinking about either removing, like New Jersey was an example. They were thinking about suspending their rabies regulations, and as a result of this study, they didn't. They realized, it's still an issue. So I don't know what the situation is for Vermont. For New Hampshire, you can see on the map that white pine is a huge resource. For Vermont, it's not as much of a resource as for New Hampshire, but I'd be interested in looking at it. So.
Yeah, certainly. I think it'd be worth a conversation. I do have one question from Earl. Is there any experience using herbicides on rabies in terms of?
Yeah, good question. Yeah, so rabies growers don't want to have disease in their. Most of the people whose houses we visited in the survey in New Hampshire weren't growing commercially, but commercial rabies growers don't want to have disease because it also affects the quality of the fruit. So there are all kinds of. They do manage for this in New York, where they grow rabies commercially. They're protectant foliar sprays, so chlorothalonil and things like that will protect the leaves from getting infected. There's horticultural oils for people that are growing them. for organic production. So there are measures to control disease on the ribes.
That's great. Okay. Well, great. Well, I don't see any other questions coming in, Isabel. So with that, is there anything else you wanted to add? Or
No, thank
before
you
we
for your
wrap up?
attention.
Okay. Well, thank you again, Isabel. We really appreciate you taking the time to share this research with us and talk to us about the white pine's status in the region. And again, just a quick reminder to folks who are participating in the webinar today, at the end of the webinar, you'll be getting a link to a survey. Please go ahead and take some time to fill that out. We really value your feedback and appreciate your feedback. So thank you again, everybody, for joining us today. And as I mentioned, our next webinar will be on March 1st, and we'll be hearing about tree biomechanics from Amanda Bruns with the University of Connecticut. So with that, have a great day, everybody, and thank you again for tuning in. Take care. And thank you, Isabel.
Thank you, Kate.